I'm Dorita Chang, and I currently I'm an assistant professor at the University of Hong Kong, and I'm also currently the program director of our undergraduate neuroscience program, which is actually about a 3-to-3-year-old program. And my main research interest is in the visual neurosciences. So I spend all of my energy trying to understand the functioning of our visual system using a lot of different techniques, primarily a lot of neuroimaging, fMRI work, MEG work, which is a spin on neuroimaging. Of course, we do some behavior that we combine with all this imaging data. And recently we're doing some biochemistry measurements with spectroscopy. We're doing a whole bunch of stuff. We do brain stimulation, so a lot of different neuroscience cool things. That's a really big question, and the answer usually surprises all of my students. So in fact, when I entered, we don't need to go back to when I was young, but as in whenever grade school, high school, secondary, whatever. So when I entered university, I entered as a science student who thought, and that's what ended up happening was I declared a biology major with a genetics concentration, and it's an interesting story there because, of course, as in any undergraduate program, you have some free credits to do electives. And with those electives I took some psychology courses, but not like psychology courses, but they were like all brain and behavior. And then I remember there was one course on visual neuroscience that was the first thing that happened to me that I found really fascinating. And then I ended up taking, with my other electives, like some of these I think it was I forget exactly the name, but it was something on neurochemistry and neurotransmitters. I found that really cool. Then I took like a lab course where we got to like slice open sheep brains, and so I think you can see where I'm going here. But my electives sort of became more interesting than my core biology stuff that was happening. Like it was so tedious for me. I think I don't know if we have anyone intending on doing a major in biology later, but I'm sorry, I hope I don't offend you, but I found it very boring. I don't know if it was the hour that the 8:30 biology classes, or it was like the stupid organic chemistry course, suffering through physics of all sorts, calculus, and it was tedious, and it wasn't. The interest kind of started diverging towards brain and behavior and neuroscience stuff. And so in my third year, which is quite late, I changed my major. And it is late, and I suffered for that by taking summer school. It's not so common here in Hong Kong, at least in Hong Kong U, but summer course credits with the equivalent classes we could take to kind of graduate on time. Of course, my parents were going to kill me if I needed a fifth year or something just because I changed my major. I graduated on time, and so that's how my interest started in kind of like the neurosciences, understanding the brain and behavior. And vision part specifically, what happened was in my fourth year, I got to do some lab work with my undergraduate, it's like capstone thesis stuff that you guys heard about. And I ended up doing it with a professor who was doing visual development. And I know it sounds funny to say this, so many years later, but I think that capstone experience was probably my most kind of life-changing experience. That's sort of why I got so interested, and I got, and that's, how by that time I decided, okay, I think grad school is next for me, and then to be in a vision lab, and then that sort of just snowballed into a career in vision work. But yeah, but by no means did I grow up liking vision, liking neuroscience. No, I think, and it's one of these things that you guys will learn as well. I think just you won't know you like it or not until you start doing it. Work both ways to just as much as I thought genetics and biotechnology was going to be so cool. And I started studying it and I just hated it. So, I'll get it to you from if I could take myself back to that year where obviously I don't know nearly as much as I do now. It was actually a behavior only lab. They didn't do any like neuroimaging stuff, but the idea was that we were going to chart through. I mean, we couldn't chart through the entire lifespan, but through a short period of lifespan, how like inhibitory mechanisms were a little bit different in children than in adults. And I remember there was, I don't want to go to too many boring details, but there are a lot of things you can do in a visual stimulus to mimic, for example, the receptive field of a neuron, or to make, okay, if we assume the neuron is paying attention to this spotlight, right? And we, make this surrounding ring carry some content that is different from the inner part of the ring, then maybe we can make some hypotheses about, okay, if they have very strong inhibitory mechanisms, maybe that content in the surrounding ring will be distracting for them in the middle, or maybe it'll be facilitating. So principles like that are obviously based on physiology data, based on neuroscience data. But then what was really cool was that we could use, sure, it was only behavior, but we could use behavior to make really cool inferences about brain mechanisms. And it's a really good place to start, obviously, definitely for an undergrad who, is still just starting to get into sciences, but just to understand, why do we do these things? And what is the link between what we have up here as a stimulus on the screen to these subjects, to literature, and then to rounding about when I have my data, how do I make those inferences back to what it means for a 7-year-old child's brain versus, an 18-year-old adult's brain? And so I felt that that was sort of why it was so life-changing. It was suddenly realizing, oh, hey, this kind of puzzle solving and this story making from your behavior data actually is a lot of fun.